So my dear children, in this activity, we'll be investigating the presence of starch in a plant leaf. So to do this activity, I have several instruments with me. So first of all, I have prepared the instrument in here by using a tripod and a mesh and our water beaker in order to heat the plant leaf. So I have a crucible I have a forceps with me, then a small bit of a forceps in order to handle these leaves, right? So I have 2 green leaves with me here, then a boiling tube, after that some alcohol, right? Ethyl alcohol, ethanol. So first of all, what I'm going to do is I'm going to take these 2 leaves and insert it to the water like this, right? Now what you have to do, you have to boil it very well. So let's slide it up. Okay, now we have to boil it very well. Right, so let's keep the system for a while until the 2 leaves are going to boil. Okay, so we have to boil it very well in order to remove the cell walls and to be open and to open the chlorophyll. So after that, we are going to put the 2 leaves inside the boiling tube. Then after that, I'm going to add some alcohol. Then we have to reheat in order to remove the chlorophyll. So with the chlorophyll, you know that we can't clearly get an observation. So therefore, we have to remove the chlorophyll in the system of the leaves. So let's keep these leaves for a while so that they would boil up and to expose the cell walls to remove the chlorophyll. So as you can see here, now the water is starting to boil. So we have to insert the leaf within the system so that the leaf also boils very well. Right, so now you can see the water has started to boil, right? So you can see the bubbles over there, and now we have to boil the leaves very well within the water, right? So you have to insert the leaves within the water and we have to boil it very well for a few minutes, right? So once again, we'll keep the system in order to boil for a few minutes. Then after that, we'll take it out and let's boil it inside the alcohol. So we'll keep it for some time in order to boil it very well, right? So now as you can see here, now the 2 leaves are well boiled. So now I'm going to take it off and place it within this boiling tube. So you can see that the color of the leaf has got changed right now, right? The color has got somewhat changed. See, you can see that it has like brownish color nature. Okay, so now I'm going to place this larger leaf into my boiling tube like this. We have to keep this turned on. You know that we have to boil it again by using alcohol. So let's insert the second one. Right. Now what you have to do, you have to add some alcohol into the boiling tube. So you have alcohol with me. I have alcohol with me right now. So I'm going to insert some alcohol. Alcohol is highly flammable, so it's wise to keep away from the fire, and we'll insert. Right. So I'm going to wipe it down. So my dear children, now don't boil alcohol directly under a flame. It's because that you know that alcohol is highly flammable. So when you are inserting this thing, make sure you insert this thing within water like this. Right, now we need to allow it to boil somewhat so that the chlorophyll will mix very well with the alcohol. So we'll allow it to boil somewhat. As you can see here now, see, the solution is turned into greenish color. It's because that chlorophyll is getting mixed very well within the alcohol solution. So chlorophyll, in order to remove chlorophyll, we need to use alcohol. So that's why we need to boil it inside alcohol. But however, you can't directly boil alcohol is because that alcohol is highly flammable. So if you directly heat it, there might be a possibility that it would, you know, like explode and you might get hurt. So you have to be very careful when you are heating alcohol. Right, so you can see now the solution has got turned into a greenish solution. So the smaller paper now has turned into like white color. So let's keep the system for a while, and after that we'll take the 2 leaves out. Right, my dear children, now I'm going to stop the boiling process. So first of all, I'll put off the flame. Right, so let's take it out and let's put off it. So before that, we have to take the boiling tube out. So I'm going to remove this system away. Right, so now what I'm going to do is I'm going to remove the 2 leaves here. Right, so let's see what has happened over here. So I'm going to use that forceps thing. Let's take somewhat up. So as you can see here, now the leaf has got turned into a white color. So we'll keep it on the watch glass. So as per to the larger big leaf, it doesn't change that much into a white color. So therefore we'll use the smaller leaf, this one, in checking for the presence of starch, as it turned into completely white color. So we'll leave it here, and you can see that so solution has got turned into a green color. So that it's because of dissolving chlorophyll within the alcohol solution. Right, so I'm going to remove it now. Let's keep it in here. Now before we are going to add starch into this one, we need to wash the leaf very well. Right, so I have a water beaker with me. So you have to place the leaf and we have to wash it very well, right? So in order to dissolve all the alcohol, so dissolve alcohol, I'm going to wash it very well. Right. Okay. Now let's take the leaf out. Now this is my plant leaf which I have prepared for the starch test, my dear children. So what are the procedures? Once again, we boiled using the normal water. Then after that, we took it out and we placed it in a boiling tube and we filled the boiling tube with alcohol. Then we placed it inside the water beaker. Then once again, we boiled it. Okay. So after that, now I am going to test this system for the presence of starch. Right. So my dear children, I have cleared my desk so that you can observe the leaf very well. So now this is my leaf which I have prepared for the starch test. Okay. Now it has a like white, like a faded color, not exactly white, but however you can observe that the color has got faded somewhat. Now I'm going to place it over the white porcelain tile so that you can observe the color change very well. So I'm going to remove the watch glass. Right, now what I'm going to do is I'm going to add some drops of iodine solution into the leaves. Right, now let's observe what's going to happen. So I have an iodine solution with me and I'm going to use a dropper also, right? So see, so this is the normal color of iodine. It has a brownish yellow color, right? Brownish yellow color. So let's add some iodine drops into the leaf. So actually you have to spread it here and there, right? As you can see here, now the color is getting changed, right? So you can clearly observe the color is changing. Now the color has got changed into purple blue color. You can see, you can clearly observe that the color of the iodine has got turned into a purple or blackish blue color. See, it has turned into a blackish blue color. So what I'm going to do is I'm going to take once again some drops of iodine to show the color difference for you guys. So check the difference in colors very well. See, this is the original color, the brownish yellow, and in here you can observe the purple blue color, right? It has a dark blue color nature, right? So with this test and from this observation, my dear children, we can finally conclude that the leaves is producing starch when there is photosynthesis. So if the photosynthesis has occurred within the plant leaf, then by only we can identify the presence of starch. So we can say that within this leaf, the photosynthesis has got occurred. That's why the color has got changed into purple blue color as per to the activity. So finally we can conclude that photosynthesis has occurred within the leaf and a starch has been produced. It's because that the solution of iodine or the iodine solution is getting converted into purple blue color or dark blue color with the presence of starch. Right, then let's head on to the classroom once again and we'll study the further things related with the lesson. So my dear children, now let's conduct the activity to show that oxygen gas is getting emitted during the process of photosynthesis. Now in here, I have prepared a system according to the diagram which is given. Okay, so at that diagram, you could remember there was hydrilla plants. So these plants are hydrilla plants. You can observe it very clearly, right? Hydrilla plants, aquatic plants, hydrilla. Then I have inserted the funnel into the system and the gas jar in here. Now instead of gas jar, I have used the boiling tube as it is very smaller so that I can collect the gases very quickly. Now if you check the bottom area around here, right, you can see gas bubbles which are around the, right, there are clear gas bubbles around the surface of the leaves, right, around the surface of the hydrilla leaves. So those gas bubbles are come and going to collect in this boiling tube, within this boiling tube. So that's why I have used a very smaller boiling tube in here. So it will take somewhat longer time to collect, at least like 100 milliliters of gas. So but the thing is that we have to collect at least 100 to 150, at least 50 milliliters of oxygen gas in order to test it. So therefore we have to wait till these gas bubbles are going to come to the front. Actually there is a small bit of a gas bubble which is created in here as well, right? At the top there is small bit of a gas bubbles, right? So like that way we have to wait till all the water comes down and the gas bubbles around the hydrilla plants come up, right? So we'll leave the system for a while, right? And after that we'll get the gas and we'll test it by inserting a lighted splinter or else a halfly lighted splinter and see what's going to happen over there. So if it is going to ignite very quickly, definitely the gas which contained in here is oxygen, right? So we'll leave the system for a while, right, like this, and let's collect the gas into our boiling tube and let's test it by inserting a glowing splinter. Okay children, now I kept the system for a while, right? I have kept the system for a while. Now you can see that the gas bubbles around the structure or around this system is very less now. There are less amount of gas bubbles now. Earlier it was like huge amount of gas bubbles could be observed around these areas. You could observe it, but now very less amount of gas bubbles are there. And you can observe at the top now, like this much of a this much of a height, the water has dropped down, right? Like this much of a height, the water has dropped down. You know that in the earlier instance there was plenty of, I mean, like to the top of the boiling tube there was water, but now water has dropped down, means that a gas has been collected on the boiling tube. So my dear children, it's difficult to insert a glowing splinter with this much of a volume. So therefore I need to wait till the boiling tube is completely getting filled with oxygen gas in here. We don't know whether this gas is oxygen or whatever the what kind of a gas it is. In order to check that thing, we have to insert a glowing splinter, right? The flame should be put off and we have to insert the splinter. Okay, then we need to observe whether the splinter is getting lighted up again or not. So we have to wait somewhat, right? So you can see that now up to this much of a height the gas has been filled inside the boiling tube. However, we have to wait till it is getting completely filled with oxygen gas or the gas which is getting emitted from the hydrilla plants. So we'll wait once more and let's see what's going to happen when I'm going to insert the lighted splinter, right? So we'll wait until this is going to completely filled with oxygen gas. Right, my dear children, now we have waited long enough. As you can see now, the whole boiling tube is now filled with the gas emitted from the hydrilla plant. So I have now I need to collect the sample very carefully. First of all, I'm going to insert my hand, right? So I'm going to take it up. However, you can see that I can't keep my hand over here as the funnel is somewhat lengthier. So what I'm going to do is I'm going to add some more water into it very carefully. Now let's try to take it. Right, now I have the gas with me right now, my dear children. Now I'm going to insert the globin splinter into it. So I'm going to take the globin splinter, right? So as you can see, there is no flame in it, just the smoke. So what I'm going to do is I'm going to quickly remove my finger and insert this thing and observe the flame very carefully. Okay, so let's see. So you can see that it has got ignited with a bright flame. So it is giving out a very bright flame. So it concludes that this gas jar or this boiling tube contains oxygen gas. That's why it is giving out a bright flame. It's because that oxygen is a supporter of combustion, right? So oxygen is a supporter of combustion. So therefore, when we insert a globin splinter by putting off its flame, right, it's going to reignite with the presence of oxygen gas, as oxygen is going to provide a support for the combustion process. So finally we can conclude from this experiment that this gas jar or this boiling tube had oxygen gas in it. Right then, so let's head on to the classroom once again and we'll write down our observations and conclusions. Okay, my dear children, now in this activity we'll be identifying how does the diffusion is going to work. Now within this activity, first of all, I'm going to use Condis crystals, right? Potassium permanganate or Condis crystals. As you can see here, these things are available as crystals, Condis crystals. So I have a small bit of a Condis crystal with me with a water beaker here. So what I'm going to do is I'm going to put this Condis crystal into the water beaker, right? So let's put another one near to it. Right, now you can see that the gradual dissolution of color within the water. Now you can see once again the Condis crystal, near the Condis crystals there are like streams of purple color which is going upward, right? It's because of now in bottom area the concentration of the Condis is very high, and in the top area the concentration or the number of particles, right? So the number of particles of Condis is at the bottom, it's very high. Okay, at the bottom you can observe it, it's very high. See, at the bottom there is large number of Condis crystals. Right? So now it's getting dissolved very quickly because of, now you can see that here in to the top also gradually coming to top as well. It's because that due to the concentration difference, right? So at the bottom there are large number of particles of Condis. So at the top very less number of Condis particles. So the concentration is greater at the bottom part. I'm talking about the Condis, my dear children. Then at the top part the concentration is low. So therefore you can see a stream of Condis. See, a stream of Condis is coming from here to here gradually, right? So we have to leave it for some while and let's observe what's going to happen after some while. So my dear children, as you can see here, now at the middle portion of the beaker, now it has got turned into purple color. A good purple color can be observed throughout the beaker, but however at the top, like in this area, there is still the colorless nature can be still observed. It's because that the stream is coming very slowly, right? So now at the middle part you can observe, right? At the middle part you can observe the purple color very well. So we'll leave it this system for some bit of a time, and once again we'll observe what's going to happen. So my dear children, now you can observe the entire solution has got turned into purple color. It's because now the diffusion process has ended. So entire solution has a uniform or else a constant composition. Now the composition means throughout the solution the number of particles are now equal. That's why it is showing a unique purple color. So this can lead us to one conclusion. Particles are automatically getting turned, or particles are automatically getting moved to a position from higher concentrated area to a lower concentrated area. So this process is referred as the diffusion process. As you can see here, now the entire solution has got changed into purple color, right? In the earlier, purple color could be observed only at the bottom. However, after some time you could observe that the purple color has completed, or purple color has taken in the entire solution. So this concludes the activity and our observation, my dear children. So let's head on to the classroom and we'll study what are the other things related to the lesson. Now my dear children, I have a balsam plant with me. Now you can see that the trunk or the stem of the balsam plant is having a green color, right? It's having a green color. So what I'm going to do here is I'm going to take this balsam plant and insert this thing into the potassium permanganate solution or to the Condis solution. So I have inserted it. Now we need to keep this thing for a while so that the roots will absorb this solution very well. Then after that, let's see what's going to happen, right? So let's keep the system for a while. Then after that we'll observe what's going to happen in the next step. So my dear children, within this activity we'll be observing the nature of osmosis. So in order to observe the nature of osmosis, I have with me a colored water setup. Now in this beaker you can observe red colored water, right? And I have several blossom plants, right? I have several blossom plants. So as you can see here, the trunk or the stem of the balsam plant is in greenish color right now. Okay, it's in greenish color. And I have somewhat a smaller one as well in here. Now what I'm going to do is I'm going to insert these balsam plants into the colored water solution like this. Then after we have to observe the two plants after like 2 or 3 hours. So let's keep the system in the same position for about 2 to 3 hours, and after that we'll observe what's going to happen or what's the change within the stem in the plant, right? So we'll leave it for some while and let's observe after some time. So my dear children, now I have kept this balsam plant for some time at this colored water. Now you can clearly observe the color change. Now the earlier this plant was in green color, greenish color. Now you can observe at the base of the plant the red color is very well observed, and at the top of the plant also there are few colors, right? Few, I mean like red color can be identified in few stops, right? So here, at here and over here also you can observe that red color very well. And within the base, like in this area, the red color is quite well observed. Now it's because that, root hairs contained within the balsam plant is going to absorb this water that has red color pigments, and it's been transported towards the xylem tissue, right? So that xylem tissue now in red color because of the red color pigments. So that's the action of osmosis process. So here the root hairs, the membranes in the root hairs is acting as a semi-permeable membrane. So it allows to flow water through the membrane. So by that way the water is going upward through the balsam plant. So that's why we can observe the red color in here, right? So my dear children, let's move on to the classroom once again, and we'll write down the observation and the conclusion of this experiment. Now my dear children, let's further investigate about this osmosis process. Now I have used here a thistle funnel fixed to the stand over here, and here I have placed a beaker. Now within the thistle funnel you can observe the Condis solution, right? Potassium permanganate or Condis solution. Now the Condis solution is having some amount of salt in it. So what I'm going to do is I'm going to pour this water into this beaker right now. Before that, my dear children, please check the level. So the level is now the water level or the solution level is now at here. It fills the bulb of the thistle funnel, right? Up to the bulb of the thistle funnel. Now you can observe the Condis solution. Okay, Condis with salt solution, right? Up to the bulb level you can observe it. This right up to the bulb level only. Now what I'm going to do is I'm going to gently pour this water into the beaker. Right. Now let's keep this system for a while and let's observe what's going to happen. At the bottom of the thistle funnel, my dear children, there is a cellophane paper, right? Cellophane paper which is firmly fixed to the mouth of the thistle funnel by using rubber bands. Okay. So if there is an entry or exit between the particles, that's going to happen only through the cellophane paper. It's going to happen only through the cellophane paper. So let's observe what's going to happen after some time. So remember, the water level was at the bulb level in the thistle funnel, not the water level, the Condis level or the Condis solution with salt. So that level was at the bulb level. Okay, it's exactly going to fill the bulb. So I have inserted some water. Now you can see the two levels of the water plus the solution in here, the Condis solution level. So let's keep the setup for several hours, and after that we'll observe the changes. Okay, my dear children, now you can observe the setup very carefully. If you observe the setup very carefully, you can see that the Condis level, now inside I told you that we inserted Condis with salt. So here that level, the solution level has raised somewhat up. In the earlier case, when I was inserting the solution, I was inserting only to the bubble level. That's it. But however, now it's been raised up to some certain height along the thin tube. And if you check clearly the color of the solution, so the color of the solution has also get reduced, right? Earlier it had a very good purple color, but however now the color has got reduced somewhat. Reason for this thing is that inside the beaker inside this beaker there is large number of water particles. But however, within the Thistle funnel, there is less number of water particles as there are more salt and condensed. So automatically what happens here is water particles is going to move through the cellophane paper towards the Thistle funnel. Okay, that's what's going to happen in here. So my dear children, let's head on to the once again to our classroom and we'll identify the further things related with our lesson. Right, my dear children, to investigate further things related to the osmosis process, here I have used a petiole of a papaw plant, right? Papaw plant leaf. So that petiole is filled with a salt solution up to the topper level in here. Okay, it's filled up to the topmost level, a salt solution, right? Then this end of the petiole is closed. As you can see, it's been closed, right? So there is no way of entering or exiting particles from this end, right? As it is sealed. Right. Now within the beaker you can observe water. Now inside the papaw petiole you can observe a salt solution. So if the particles are going to exchange between papaw petiole or with the solution or the water in here, there is only one way. That way is through this end, right? So let's see, we'll keep the system for a while and let's see what's going to happen with our system, right? So you know that if the papaw petiole contains salt solution, means that the water concentration within the tube, papaw tube, is very less. Water concentration is very less as it contains more salts. Within the beaker there is large number of water particles. So according to the osmosis process, what should happen here is water should come towards the papaw tube and it should drop off from this end to our board or to our desk in here. So let's see whether this thing is going to happen or not after few hours. So as you can see here, now the solution within the papaw tube has come to the very top and you can see that the water drop is coming down. See, there's a water drop coming down towards the papaw tube. You can observe it very clearly, right? So it concludes that water is going to draw from the beaker towards the papaw tube and it is coming away from this end. It's because of the osmosis process that's going to happen within the papaw tube. Papaw tube is allowing only water particles to enter to the system, right? However, the salt solution is not getting entered to the water in here. It's because of the difference between the number of particles. So water which is getting drawn into the papaw tube come towards this and it is getting expelled from the other end. So if you're going to keep this thing for a longer time, you can get a more closer observations. Right, my dear children, now in order to show that the transpiration is going to occur in a plant, what I'm going to do here is I have taken a potted plant. Now I'm going to take this polythene bag, right, and I'm going to cover up one branch that has leaves like this by using the polythene cover, right? Then after that I'm going to seal it very well, right? After that, I am going to seal it very well. Right, like this way. I'm going to seal it by wrapping a piece of thread like this. Right. Now what I'm going to do is I'm going to keep this system, right, for few hours. If you can keep it in the sunlight, so by that way you can get more quiet and faster observations. But however, I'm going to keep this system in an environment, in an open environment, right, for about 2 to 3 hours. Then what I'm going to do is I'm going to observe what are the changes that has occurred within the polythene bag. So let's keep the system in an open environment, and let's observe what's going to happen. So my dear children, now I'm going to remove the system. Now here, actually I'm going to remove the polythene bag in the system. So here you can observe several droplets. Within the polythene paper, you can observe several droplets here, right? So to check what are these droplets, I have a chemical substance here with me. So this is the same chemical substance. The blue color one, you know obviously what is this blue color one. So it's copper sulfate. But however, this is hydrous copper sulfate, which is in blue color. Hydrous means when absorbing water vapor in our atmosphere, this kind of light blue color or like white color copper sulfate is getting converted to a wonderful blue color like this. So what I'm going to do here is, in order to check whether these droplets are water or not, I'm going to add this anhydrous copper sulfate, not the hydrous one. I'm going to insert the anhydrous copper sulfate, right? So let's add some anhydrous copper sulfate and see whether the color is getting changed into blue color or not. Yes. Now if you check the system carefully within here, you can see that the droplets are getting converted to good blue color. It's because of the copper sulfate, it's because of the water which is present within the polythene bag over here. So if I'm going to add more, you can see the color change very, right? See, you can observe the color change. So here you can observe the color change very well, right? So it's now near to this color. Check my children, it's very near to this color, right? So actually there are a small bit of droplets in here. So I'll take these into my hand. See? So it's equal to this color. So anhydrous copper sulfate is turning into this kind of a blue color from this color, light blue, to this kind of a blue color is getting converted. Oh, we can observe this kind of a blue color in copper sulfate solution only with the presence of water. So this is called as anhydrous copper sulfate, while this is called as hydrous copper sulfate. Anhydrous copper sulfate is getting converted to hydrous copper sulfate with the presence of water. So we can say that this polythene bag contains water droplets. It's from the chemical change which is going to occur within anhydrous copper sulfate. So my dear children, it's obvious to us that During the transpiration, water is removed from the leaves as water vapor. So that water vapor is getting condensed within the polythene bag to form water droplets. So now let's head on to the classroom once again and we'll discuss further things related with our lesson. Right, my dear children. Now to observe what happens during the transpiration process, I have prepared this setup. So in this setup, you can observe I have taken a branch of a plant, right? And I have inserted to the cork over here, well sealed, right? So the whatever the particles is not entering into the system or not getting outside. So within the U-tube, you can observe there is water. So I have sealed it very well so that I can guarantee you that water is not getting evaporated from the system. At the other end, I have now I'm going to put for the other end some amount of coconut oil, right? But however, I need to explain the experiment, so I didn't put that thing still. So this is my setup. So in here, altogether you can observe water, the branch of the plant, then firmly fixed cork within the U-tube, right? And as it is firmly fixed, none of the water particles are getting exit from the system. The only way of exiting particles is through the plant branch only. So what I'm going to do is now I'm going to seal the other end by inserting some amount of coconut oil. Right, so I have sealed it. Now as you can see, if you observe this end very carefully, right? Now the water level is at the topmost level, like 1 or 2 centimeters behind the top. So it's at the very top, like here means like 1 or 2 centimeters behind the topmost level. Water is like up to this kind of a level. So what I'm going to do is I'm going to keep the system for a while. You know that now this system is very sealed as it is not being exposed to environment. So only way of exiting water particles is only through the plant leaves, right? So let's observe, let's keep the system for a while and observe what's going to happen for that water level at our other side, not this side, at our other side. So let's observe what's going to happen over there after few hours. So let's keep the system and we'll observe after few hours. So my dear children, as you can observe, after few hours, the water level within our other side, not the area that has the branch, but the other side, the water level has dropped down. It's because the water is getting absorbed by the branch of this plant and it is getting vaporized or evaporated to our environment through these leaves. So that's how the transpiration is going to work. So upon evaporation of water, the water level is going to decrease gradually. That's why at our left hand side, the water level gradually going to come down. So you can clearly observe now in the earlier case, the coconut oil layer was at the top, right? Now there is a clear gap between, right? Now there's a clear gap between the coconut oil layer plus the edge of the U-tube. So my dear children, this concludes that during the transpiration process, water is getting absorbed and it is evaporated to our environment. So let's head down to the classroom once again and we'll continue with our next works.